and gentlemen, let us welcome opening and welcoming remarks from Dr. Vatu Papu Zamxaka, the Deputy Vice Chancellor for Research, Innovation and Engagement at the Tshwane University of Technology. Over to you, Doctor.
Thank you so much, Program Director, Mr. Mosase. Good afternoon, colleagues. I hereby wish to welcome you all on this very important webinar. As the Program Director has alluded, the webinar is dealing with a very important matter within our society at large, the issue of vaccines. In particular during the time of COVID-19. The webinar is looking at challenges and opportunities for vaccine development. And as the university, we're looking forward to be hearing from all the experts, their own perspective, the experts that are coming from the pharmaceuticals, the health professionals, and our own African leaders, as far as what are the challenges. Whenever there is a challenge, there is an opportunity that is being created by that particular challenge. And all our eyes are looking into our scientists to come up with innovative ways of vaccine development. So as I welcome you, I am also saying to ourselves, we are the ones that we've been waiting for. I'm particularly excited. Yes, I'm the Deputy Vice Chancellor for Research, Innovation and Engagement, the TUT. But as training, I'm a scientist. So I'm particularly excited when I see the fellow scientists taking the lead in solving the problems of the world, because science, it is to solve the complex issues of our societies. So for this day, unfortunately, I can't stay longer as much as I wanted to stay longer to hear what the colleagues are going to be putting on the table and what they will be implementing. It's not only about us hearing, but it's also about the experts implementing that which is important. So on behalf of Tshwane University of Technology, I would like to officially welcome all of you. I am truly excited at heart. I've spent the past 6 years in clinical research, so you would understand why I'm particularly excited when topics of this nature come to the fore. So colleagues, without any waste of time, we would want to really open the floor, and deliberations needs to take place, and the deliberations that are accompanied by action. So we want to see, in particular, Africa taking the lead. I must also comment as I close and say it was really sad, and I don't want to say embarrassing, but it was not nice to see that for the vaccine for COVID-19, as Africans, we didn't take the lead. So I believe that with webinars of this nature, my fellow Africans would represent us and take the lead, such that when things like this pandemic, like this one, do befall us, we are seen to be in the forefront of bringing the solutions. So let us be part and parcel of the solutions that are needed by the world over. So I am really looking forward. I see that the session is recorded. So I will definitely have time. I had to step out of another executive management meeting to come and welcome you. So I need to go back. So my sincere apologies that I can't stay longer, but I'm glad that it's being recorded. I can assure you that I will be listening, and I'm looking forward to see the implementation of that, which will be the resolutions that will be taken in here. The time to meet and talk and we go is over. We need to meet, talk, and take some resolutions that need to be implemented. With those words, I thank you, ladies and gentlemen, and I welcome all of you on behalf of our university, Tshwane University of Technology. I thank you.
Thank you so much, Dr. Zamxaka. And I would also like to acknowledge the chairperson of the Tshwane University of Technology Council, Mr. Tilson Manyoni. You are welcome. And also acknowledge in absentia, because we did get the message from the TUT Vice Chancellor, Professor Lawrence van Staden, the executive management of the university, the student leadership and its leader, who leads them in this session today, Mr. Mike Mutiba. Thank you so much for joining us today. I would take it that at the beginning, the remarks of the program director have already been made. So chasing time so that we can finish as scheduled, I would go straight to introducing our first speaker for the day, and that will be Professor David Katerere. And I will read very slow because there are words here that I'm seeing for the first time in my English vocabulary. So Professor David Katerere is the Research Platform Chair of Pharmaceutical and Biotech Advancement in Africa at Tshwane University of Technology and Chair of Pharma Connect Africa, a non-profit organization which is a premier convener of the University of okay. Sorry about that. It's a premier convener of the pharmaceutical industry in Africa. He holds a PhD in pharmaceutical science from the University of Strathclyde, Scotland. He has worked in pharmaceutical and biotech practice and research in the past 25 years in 4 countries on 3 continents. He has several inventions to his name, including the nutraceutical Nicelo and COVID Connect app for use in clinical trials and post-recovery. He has published over 50 journal articles and is co-editor of 3 books, Ethnoveterinary Botanical Medicines, System Analysis Approach for Complex Global Challenges, Springer 2018, and Traditional and Indigenous Knowledge for the Modern Era. He is a member of advisory committees of SAPRA and the Global Health Supply Chain Consortium, co-founded by faculty at University of Michigan and University of Southern California. Professor Katerere teaches and researches across the pharmaceutical and biotech value chain, including product development and clinical testing, substandard and falsified medicine, vaccines, and medicines governance. Thank you so much, Prof. And I hope I did justice to some of the words. If not, I'm immediately enrolling in your program in the following week. Over to you, Prof.
No, thank you very much. No, you did a great job, and thank you for inviting me. So yeah, good afternoon. So I'm here to just talk about the short history of vaccines, if you like. And maybe just to introduce what the chair is about. So the research platform chair really works across the value chain of the pharmaceutical industry. And these are the thematic areas that we are interested in. So regulatory strengthening, tech transfer and support, research and development, and of course, business and business support. Because even for the vaccine issue, it's because there is no business case in for any one country actually, which is one of the biggest barriers for producing vaccines. And interestingly, this time last year, before the lockdown, we had already started talking about a workshop, mostly on biosimilars, but also with vaccine development. So we are working towards that workshop with ICGEB, which is then aimed at capacity building, regulatory science, and also a pilot manufacturing, as just a small contribution to this whole thing. And these are the various partners that we are working with. So to the business of the day then. I think to understand the history of vaccines, we need to understand the history of disease. And disease starts with an outbreak, which is a sudden rise in cases and in a very small area. So let's say typhoid, and it's really limited. And in South Africa, we've had outbreaks of typhoid, for instance. And then if it spreads a little bit and within a very short period of time becomes of a larger distribution, it then becomes an epidemic. And we've had lots of epidemics in history, but many more in probably the past 60, 70 years. And this just gives you an idea of the epidemics that we've had, from pretty much all over the world. So we've had Ebola, which we are all familiar with, and then in 2002 we had the first SARS. So actually, COVID is called severe acute respiratory syndrome 2. It's the second version of it, and in a sense that's what explains this very quick development of vaccines, because back in 2002 people already started working on vaccines for SARS. And then you would have heard stories about bird flu, the different types of bird flus, Middle East respiratory syndrome, and these were far, far away from us, so they never really made much of an impression. So out of an outbreak we had epidemics. Now in Wuhan, 2019, there was an outbreak which became an epidemic. And for whatever reason, some say because of political issues, it was not controlled. And what did it become? It then became a pandemic. In other words, a disease that affects a large geographical region. And in this case, it's not just a regional pandemic, but a global pandemic. And we've had pandemics, many pandemics. And actually, if you look at how pandemics have shaped history, you will, Professor Tinyuko Maluleke was recently saying that, places like Soweto, for instance, arose because of the pandemic in the flu, the Spanish flu. At least the first settlements that happened in what we now call Soweto came out of that, as Johannesburg CBD was being cleared of people as a control measure. And so we've had all of these pandemics leading to loss of life. And in our own lifetime, we know what HIV has done. We've lost friends, we've lost relatives, and it's been a really big pandemic. So pandemics are not in the past, they are present and dangerous, and they are also in our future. Because as you can see here, we've actually had probably more pandemics in the past 60 years than previously. And that is really as a result of globalization, travel, ease of travel. And the fact that something that happens in a place that we had never heard of, Wuhan, can sweep across the world in a matter of weeks. Okay, let's then talk about vaccinology. And I wanted to call it with the Zulu, isiZulu name, izinkomology, if you like, because actually vaccine, the word comes from vac, which is French, Latin for cows, because we are told in history that Dr. Jenner was the person who discovered vaccine. And this by observing milkmaids, the fact that milkmaids were actually not prone to getting smallpox infection. So here's a picture, and this is what a vaccine has grown to be. So back then they would just take an extract from cattle and probably just inject it. But we know that a vaccine has all of these other things. Some of these then make it potentially toxic and also sophisticated. But that is the history that we know. There is a hidden history, an untold history of vaccines. And it starts with what is called variolation, which is really just making incisions on people's bodies. And the earliest recorded is Chinese. There is no doubt that in other traditions across the world, this was happening. But of course, unfortunately for us as Africans, we did not record anything, which is why I am very specific about writing things. So, and before 40, 60 years before Dr. Jenner's discovery, as it were, there was actually a slave called an Anismus who had been transported from West Africa. And in the outbreak of smallpox in and polio, I think, in the Americas, he had then actually talked about how they controlled pandemics in West Africa. And his slave master at that stage was fairly enlightened in a sense and started experimenting. And therefore, they ended up actually saving many, many people in the US. This is untold history. And why is this interesting? I myself did not know this until recently, but however, in 2010, when we put together this book, our first book, which was on ethno-veterinary botanical medicine, one of the authors here actually pointed to the role that a vaccination plays among the Fulani herdsmen in Nigeria. And in that case, what they would do one of two things when they had a disease that was killing cattle, and of course it would be an outbreak in that case. So they would either take the lungs of the dead cattle and make incisions into those that are alive and put in there. So the variolation that we talked about, they would then put in there this extract from the lungs. And in that case, they are actually vaccinating their cattle. The other thing that they did is that they would get cattle to go downwind of those cattle that are sick and then be exposed to small amounts of the whatever agent was causing that disease, and in that case then be vaccinated. I would hasten to say that and it would be interesting to look maybe at the Zulu traditions, because wherever you have cattle, you have ways of managing that. So probably among the Zulu, probably among herdsmen in East Africa, there would be ways of vaccination. But there's an inglorious history of vaccines that we cannot really run away from, and I would like to briefly talk about that. Okay, so with vaccines then come laws. It becomes compulsory for people to be vaccinated. And this is from 1907 in England, when men were actually sent to prison because they refused to have their children vaccinated. So there's always been fear around vaccines. Of course, now we give it a name, anti-vaccine, anti-vaccine movement, and social media has just kind of made it worse, I guess. But here's a headline from the UK today, where they are saying it will be compulsory for care home staff to be vaccinated. In Africa, we have heard in some countries threats that if you do not get vaccinated, you will not be using public transport. Well, it's already happening. So in Israel, you cannot use public transport unless you show a vaccine passport. What are the ethics of these kind of frameworks? But you see, when you balance, you have to then balance individual freedoms with public health priorities. So I grew up in colonial Africa, and we had our own form of passport. So this is BCG, and you will find that across most of colonial Africa, people would have these marks on their bodies, and these really came from BCG. The interesting thing is when you look at South African arms, they tend to have not so marked a form of BCG, whereas in some colonial countries they made sure that this was very visible. The reason why this was is because there were two things when you went to school. They looked at the mark to show that you were vaccinated, and number two, they looked at your height. So if you were too short, you would not go to school. If you did not have a mark such as this, you would not go to school. So the whole issue of vaccine licenses has been going on for a very long time. The other issue is vaccines have the potential to cause harm. And recently, and I'm a Rotarian, so recently Rotarians have been very proud that they were involved in kicking wild polio out of Africa. Yes, it's been kicked out of Africa. However, a type of formulation, which is the oral form of polio vaccine, has now been reported to potentially cause polio in itself. So the issue is that vaccines are made from weakened, if you like, agents. So if in this case a polio virus has been weakened to such an extent that when it gets into the body of the person, it then produces antibodies. However, it could potentially mutate, right, into a stronger form of this weak virus, which can then go into the environment. And those people that are not vaccinated could potentially then be exposed to it, and they themselves would then get polio. And there are reports of this happening in Sudan and Chad. Now, here's the issue. At the moment, we have one vaccine which uses a attenuated coronavirus, and that is the Chinese one, Sinovac. The question is it then possible that it will benefit people who directly take the vaccine, but expose people who are not taking the vaccine? That is for science to answer, but vaccines have the potential to harm. Well, vaccines have the potential to kill, and science kills, or not enough science kills. And this is Sanofi. So Sanofi is probably one of the biggest pharmaceutical companies in the world. They put together a vaccine for dengue fever, which is a lot like malaria, if you like, because it's also transmitted by mosquitoes, mostly in Southeast Asia. And it ended up a campaign then in the Philippines, where they gave this to 180,000 children, ended up with 130 fatalities, which was unacceptably high. But that's because there was no understanding between what are called villain and hero antibodies. So not all antibodies that your body would produce after a particular vaccines are useful. In the case of the dengue fever, some of those antibodies kill right away, but it's very difficult to actually test these without using them. So they actually test these in animals and then say, okay, this one has will produce the hero antibodies. This one will produce the villain antibodies. So unfortunately, the problem is that science sometimes needs time, but it also needs to, you also need to actually test things out. So it's a catch-22 situation. So yes, vaccines can kill. The other issue, which is rather controversial, is the issue of vaccines and HIV. And I've been very careful to put here a book which is worth reading, an amazing book, which says that HIV might have inadvertently come through from manufacture of poliovirus. So in other words, HIV was a contaminant because in the 60s, a lot of work was being done in the Congo, what we now call Zaire or the DRC Congo. And for us to actually culture virus, we need to use mammalian cells. So in this case, they were using cells from chimps. And these cells from chimps had what is called simian immunovirus, which is very close to HIV. And you can see here there's a tree that shows just how close these simian viruses are. And these would have then inadvertently ended up in cell cultures and ended up in polio. So that is the one theory. The other theory is that there was also a lot of needle sharing back in the 60s, 70s. So once you had people who might have had HIV or even other diseases, then that would have spread through the population through mass vaccination. So certainly with BCG, you know, they didn't change needles. In the old days, there was no need for changing needles at all. In fact, sometimes they would sterilize these needles by boiling them. My father was a healthcare worker, and you know, we actually used to boil needles in between to sterilize them. Right. So that is in the inglorious history. But let's think about the glorious history of vaccines. Just to be balanced, what we know is that vaccines save lives. Many of us would not be here today if we had not actually been given vaccines. So let us look at wild polio, for instance, which I've talked about, and where Rotary and Bill and Melinda Gates have played a huge role. Right, so from 1988 to 2015, we had the last case, actually. So everything came down here from thousands to just one by 2014. And by last year, 2020, we had eliminated wild polio as a result of vaccination. Nothing else would have eliminated wild polio. No treatments, no therapeutics. The other thing is when we look at measles, for instance, just over these past 15 years here, I guess we would have to go to the past 20 years, but I didn't have that data. But there's been a 79% decline globally, leading to over 17 million deaths being averted. As you know, measles is one of the big killers of children. But as a result of an intervention such as vaccines, we see that huge change and the improvement in economies, the improvement in quality of life. What we also know is that there's a huge return on investment in vaccines. So here's something from Gavi where they say that if you put $1 into public infrastructure, for instance, your return on investment is only $3. If you put $1 into treatment of cardiovascular disease, it's only $5. But if you put your dollar into vaccines, you're going to get $16 in return. So in other words, you are eliminating a lot of disease, illness. You are eliminating people ending up in hospitals because they are sick, but you're also then eliminating disability, lost years, you're actually increasing the economic potential of your population. So to conclude then, what can we say? We can say, like every human invention, vaccines are not without fault. So if you look at, there were no road accidents until cars came along. And in fact, one of the most famous scientists, Pierre, I forget his name now, Pierre Curie, so Marie Curie, he was actually killed by a wagon, right? So if there had been no wagons, there would have been no loss of life. So with every advancement, there are obviously faults. However, what we can say is that vaccines in general have saved more lives than they have killed. But to improve the efficacy of vaccines and also the outcomes of vaccines, we really need good science and also ethics to ensure that there's quality. And that's why a workshop such as this is important, because it means that we are now thinking about how do we train people in regulation, for instance, for better regulatory oversight, but also how do we train them to become more competent at making vaccines. It's not rocket science to make vaccines. The other thing is public education to understand, and most importantly, to report side effects. Because if we have a population that is fearful and uneducated, then there's a lot of rumor mongering, a lot of fake news, but also the truth will never really come out. So we would like members of the public, when they do get vaccinated, to report whatever type of side effects they are experiencing. The other thing that goes with public education is that our politicians should stop intimidating people. I know that it has happened in other countries. We actually need to use the science of persuasion, and to actually say that vaccines are for the public good. In a sense, that's why vaccines don't make money for people. Perhaps a COVID vaccine is going to be the first $1 trillion investment, because obviously, the numbers of people that will have to take it will be enormous. But if you think about measles vaccine, childhood vaccines, they are really just a public good. In educating the public, we also need to use traditional healers and traditional leaders, because they are also healthcare workers. We need to use churches, we need to tap into the whole taxi network as well. And then, of course, we need to think about innovations. On what we've done on our side is that I have a student, actually a former student who works in the industry, and we're coming up with an app that's called Smart ADR. We have looked at the ways in which side effects are being reported, and we believe that it's not being done in a way that is objective, but also a way that reaches a lot of people. So we have this Smart ADR, which will be translated into many different languages and be highly infographic, so that it allows everybody who then gets a vaccine will register on the system and then be able to report ADRs. And that will also mean that if you have different vaccines, people can report to a central space, because at the moment it looks like this is all being driven by the different companies. And so I'll end. I'll thank you. And I've done a little bit of something unusual. I'd like this to be shared. And when it's shared, you can go and read up some of the references in here, so that we really understand the history of vaccines. Thank you very much, Program Director.
Well, thank you very much, Prof, for the comprehensive presentation. And I definitely learned a lot of things coming from that presentation. And I'm so grateful that this is actually being recorded, and will always be able to follow that through. And let me also acknowledge all our participants who are joining us live from different social media streams. including those that are on YouTube, those that are on Facebook, and those that are joining us listening from Tshwane FM 93.6 and TUT FM 96.2. You are all welcomed to this important webinar. Without any waste of time, I'm going to slightly alter the program to accommodate a guest who has to quickly leave for an interview with ENCA. And I have to ask that we allow Mr. Joshua Maponga, who's a popular motivational speaker, social entrepreneur, and musician. The renowned personality is also a philosopher and leader. He is passionate about community development, which has led him to work with several big organizations. He has worked with Visio Design House and Entrepreneurial Development South Africa. Joshua has also worked with the organization in the United Kingdom, Global Management Center. He is also a popular face on television and appears on DSTV channel 331 on the program Joshua. The speaker has managed to make a name for himself in the industry. Joshua is among the most sought after personality for his knowledge and thought on the principles of life. And I do know that he is also a very good instrumentalist. He played different instruments at the very same time. Mr. Joshua Maponga, over to you. The time is yours now.
Thank you. Thank you very much for awarding me the opportunity to be on this platform. And many gratitudes to my co-presenter who has just finished a very elaborate academic presentation on the history of vaccination, and Professor Katerere. He may very well see from my surname, Maponga, that Professor Charles Chiedza Maponga, who is in Zimbabwe, the head of the pharmacology and toxicology department, both at University of Zimbabwe and Buffalo University in the United States of America, is actually my uncle. So I grew up in a house where we have produced also one of the top pharmacologists in the world, with many students out of his hand. And having said that, I have been asked to zoom into the propaganda communication around the corona COVID-19 pandemic. This is not happening in a vacuum. And the weakest link on African development has been the academic institutions. And particularly, I'm so excited because I'm talking to Tshwane University of Technology, TUT, where you are supposed to be spearheading technology itself. The unfortunate part is that our universities have been manufacturing yards of colonial students who constantly come to school not to become innovative, but to become employees of various organizations. I don't see a reason why TUT cannot align itself and move its inventions in indigenous knowledge systems and technology, inclusive to indigenous medicines that are not very far from TUT. Just down the road there next to Marabastad, there are all those old ladies who are selling herbs on the sides of the street. If we can take those herbs and actually move them into the classroom, we may find that the solutions we're looking for are close to our eyes. And I'm very grateful to what Professor Katerere said, that vaccines actually kill. And that is a very strong point. If they are not administered with knowledge, if they are not administered in understanding of the body in which they're going to be inserted into, and the correct research is being done prior to the implementation. Research has shown us, even history has shown us also, that the Aborigines in Australia were exterminated through vaccinations. We also know extensively that the Red Indians in America were exterminated through vaccinations. And we also understand birth control being a factor on the African continent, contraceptions, various types of contraceptions being brought into Africa as early as the 1950s and 60s and 80s. The side effects of those contraceptions, being obesities, high blood pressure, and some of the histories that don't want to be spoken about, infertility, sterility, and the rest of it, that the list goes on and on. And what fuels propaganda, what fuels conspiracy theories is a mixed history that is not honest and truthful. Because if you go right now to the protocols of the United Nations, the protocols of the Africa Union, the protocols of the NATOs, the protocols of the COMESAs, the SADC regions, in terms of HIV, diseases and vaccinations and world population control, you will find it on paper that as early as the 1970s and 60s, people were already projecting that the population is growing faster than the resources are growing. We therefore have United Nations official papers that will be telling us to the fact that they need to start organizing and working on a strategy to reduce the world population. And knowing what syphilis did to America, for example, and knowing the relationship of the colonial system to Black people, it is a fact which cannot be run away from, that the colonial system actually sees the African person and poor people as an inconvenience to their own wealth, to their own, resources. They actually think that the African resources belong to them because they know what to do with the resources, and the Africans don't know what to do with their resources. So the African population is a nuisance that is sitting on top of world-demanded resources, diamonds, gold, platinum, and etc. And for the longest of time, the concept that HIV could have been here much longer, and the same Western world and pharmaceutical companies have not shown such vigorous interest in exterminating the disease. But when Corona comes and COVID comes, there's an excitement, an overwhelming excitement, even to the extent of forcing governments to accept the terms and conditions of donor funding. And this, so it brings me to a very complex conclusion that why are we talking vaccination? Why are we not talking treatment? Because vaccination and treatment must be discussed in the same ambit, because you treat a disease because you understand how the disease is functioning, and rightfully so, noticing that the first wave of Corona, the second wave of Corona, and the third wave of Corona, and apparently there are many more waves that are coming. The vaccination that you are giving us, which variant is it looking at? Is it the first variant, the second variant, or the third variant? This, for me, this information, therefore, fuels what you might want to call conspiracy theories, where the Black nation feels that the European community, the colonial community, the pharmaceutical community actually has an agenda of exterminating the African population. And this agenda has not become a theory in the sky. We have had to fight in Africa here for the same democracy they tell us is the best thing Africa can ever have. We have gone through various wars. Congo is still in flames to date for the resources that are there. And therefore, when you begin to look at conspiracy theories around the Corona concept of the 666, the genetically modification of these drugs and these vaccinations, honestly speaking, the African countries need to take these vaccines into their laboratories also. Look at them, check on them, verify them, look for indigenous variants, look for indigenous active substances in these chemicals, so that we can also come up with our own African-based vaccination. And having said that, to add some few notes on my professor who has just spoken, Africans have been doing vaccinations for the longest of time. And I grew up in the village where if one child had measles, our mothers would take us and put us in the same blanket with the one who had measles. Why? Because we were healthy, so they exposed us to measles so that our bodies could fight the measles. I grew up in the 60s and 70s where I was not only breastfeeding from my mother, I was also breastfeeding from 2 and 3 of other mothers who were in the house. Only now do I learn that cross breastfeeding actually improves the immune system. And to conclude my presentation, when a child is born, after some 3, 4, 5, 6 weeks, the mothers would go to the river and collect the mud from the river, put it into a dish, go to the kraal, collect the older cow dung and put it into the dish, collect a chicken and hang it on the side. When it is dead and it has worms, put the chicken into the water there. Collect some water, pour it in there. Then collect some soil, pour it in there. And then mix up this with some herbs and some salt. Then they take a baby and then they put it into this mucky, clumsy, very, very dirty substance. And this baby then swims into this thing for a couple of hours. The baby will get a bit feverish and a bit sickish, but the mother is there to breastfeed the child back to full health. But this is the most traditional, ancient way of vaccination, because the cow dung that we are putting here, you're going to be eating the cows in the kraal. The mud we've put into this water from the river, you're going to be drinking from that river. The dead chicken that is here represents the chickens you're going to be eating. And the leaves over here, these are the trees you're going to be eating. So we introduce, as Africans, the various bacteria in the environment which a child will have to live under. And so that by the time they begin to live their lives, the water they drink, the chickens they eat, the meat that they eat, and the milk that they drink from the cows and etc., their bacteria, the active bacteria are already in the body. This is me as a primitive, uneducated African who is just making noise. That we, in as much as the Western world says they are bringing vaccination to Africa, we have been vaccinating for the longest of time. When you get to a new place, my grandmother would tell me, please scoop the dust from the ground and lick it. You know, scoop some dust and lick it. So introduce your body to the bacteria of that land. It has been a tradition amongst Africans. First thing you do is lick the dust and drink the water. So the conspiracy theories we're having is because what Corona is doing is bringing a totally different variant as to how governments and how pharmaceutical companies are working with. Worse still, when you find it written in black and white, that pharmaceutical companies have absolved themselves from any side effects of this drug. Why? Because they did not have enough time to test it and see its efficacy on the human bodies. And already it is being used on human bodies. And when you talk information like that to some people who have gone to school and studied 1 or 2 degrees, it leaves a lot to be desired. What we wish for the African governments and our own academic institutions to do, it is to go through the normal process if possible. Test these things, verify them, make sure and make that process as transparent as possible. Education is important. And what other forms of indigenous methods can we use to deal with pandemics? Now that speaks to government. Disaster management strategies. Inclusive to that, it is how do we manage not only rain and floods, but how do we manage diseases that will be coming out in the future? And when we have billions that we can put in vaccination, I conclude by saying, can we take the same billions and move them into treatment and let Africa begin to find its own
I believe we have lost Mr. Maponga. Can I get an indication is if everybody else is here with me?
Yeah, we have lost him.
Okay. I guess let's call it a day for Mr. Maponga as well, who has to rush to the television interview at ENCA and move straight to our next speaker. Dr. Eugenia Barros is the director of biotech startup company, Eugenia Barros Associated Biosciences and Consulting, that applies molecular biology and other biotechnology, agricultural and pharmaceutical industries. She holds a PhD in microbiology from the University of Cape Town, South Africa. She has worked in the biotechnology program for the biosciences unit of the CSIR for 30 years. She has over 25 publications in peer-reviewed or referred journals. She is the author of 4 book chapters and editor of 1 book HIV infection, impact, awareness, and social implications of living with HIV/AIDS. She is a member of the Advisory Committee for Genetically Modified Organisms for the Department of Agriculture, Land Reform, and Rural Development. Without any waste of time, let me allow our next speaker, Dr. Eugenia Barros.
I'm trying to share my screen.
You may go ahead, Doctor.
Yeah, I was just trying to get the screen being shared. You just put this in the slide. Okay, so I'll be talking about the technology behind vaccines, and in the outline of my presentation is I'm going to be talking about the SARS-CoV-2 genome that we all know a little bit about.
Doc, may you please start your video? We can't see you.
Oh, because I already started it.
Awesome. No, you're fine.
Can you see that now? Okay.
We can see you. Yeah.
But I don't think I am able now to move the slides.
If you go below on share screen, it should allow you.
I lost my share screen now trying to do this. I lost the share screen and it's very, very big now.
Okay, it's a green button below there. Do you see it?
No, there's nothing in here now. Okay, I can see it. Can you see my slides now?
Not yet. You must go to press share and then share, then allow it to share.
Because I did that and okay, I'm going to start again. I'm going to say share. Can you see it?
No, we can't. Maybe what you can do is you can email it to me, and then I can share from my side.
Okay.
It's David here.
Yes, but for me to email it to you, okay, let me just do it. Just open the email, David.
Do you want to send it to Gmail? If you're going to email it, you can send it to my Gmail address. Okay, I've got it, so I'm going to put it up now.
Okay. Is the sound all right?
The sound is fine. Thank you.
Okay, fine. Thank you so much. And I'm sorry about this. Okay, so I'll be talking today about the technology behind vaccines, and I'll give you can I go to the next slide? Okay, so the outline of my presentation is I'll be talking about a little bit the SARS-CoV-2 genome causing the disease COVID-19 and the response of our immune system. Then the types of vaccines and technology that have been used to develop them. And also the last topic is the vaccines for SARS-CoV-2 variants. Now, before I start, yes. So the SARS-CoV-2 virus is a new strain of the beta coronaviruses. Is a single-stranded RNA virus. It's got a crown-like shape. We've got little spike proteins. So this virus contains 16 non-structural proteins. And those proteins perform various functions like a genome replication, the induction of cleavage of the host messenger RNA. Now, there's many, many functions that are essential for the virus, for the viral life cycle. But it also has 4 structural proteins. The important ones for us are the spike proteins, the nucleocapsid protein, the envelope protein, and the membrane protein. Now, before I go into more details of the vaccines, this virus causes the disease COVID-19, coronavirus disease 19, and when the virus comes into contact with us, it enters the body by attaching its spike proteins that you see there to the proteins that we have in our cell membranes, and once inside the cell, it multiplies, producing new viruses and eventually in the cell. However, our body responds to the infection of the virus in 2 different ways. First of all, we produce antibodies, and now I just don't want to come to that one yet. I'll come there just now. This is just a basic understanding of why we're doing these vaccines. So 2 different ways. We produce the antibodies that are proteins that circulate in our blood, and when they are produced, they prevent the virus from entering more cells. And the second way, which is triggered by once the antibodies are formed, is the activation of cellular immunity, where the cells called lymphocytes that also circulate in our blood, they kill the infected cells and thus eliminate the virus. So this is a very simplistic way. Now, in the next slide, please, David, we'll see in detail what we did there. So we've got the virus entering the body and how our immunity is developed. This is very detailed, all the different aspects, and this is how the immune response takes place. So in other words, what we want to do with the vaccine is vaccines do not treat, do not cure. They mainly are vehicles to enable our bodies to prepare for a possible infection of the virus. So the vaccines expose the body to the virus that will not cause disease, but just provoke the immune system. So now I'm going to be discussing the next slide, please, David. Okay, so now you can see how the different production platforms have been used. And why do we have so many? It's because there are many knowledge gaps in the immune responses elicited by the SARS-CoV-2 virus. Therefore, it is important to explore all possibilities in developing the different types of vaccines. Now, the majority of the vaccines have used the spike protein. As you can see there, the replicating viral vectors, all of them there on the right-hand side, non-replicating recombinant proteins, virus proteins, all of them. DNA-based, RNA-based, plant-based viruses. And they use the spike protein because the spike protein is a large multifunctional transmembrane protein and plays a very vital role in the attachment of the virus, the fusion, and the entry into the cell. Now, as we have all these spike proteins vaccines that are being used, those are called recombinant vaccines. And why? Because we use a little piece of a genetic of the virus and use genetic modification techniques where we insert that small portion of the virus into a vector that is then incorporated into the vaccine to trigger an immune response. The ones at the bottom, they've got a whole virion. These are the live attenuated vaccines and inactivated vaccines. So now I'm going to be discussing in more detail what these vaccines. The next slide, please. Okay, so now we start with the virus vaccines, where they use the whole virion. The whole virus is either inactivated by heat or chemical products, or is weakened, whereby the genetic material is mutated through a series of passages through animal or mammalian cells to make it less able to cause disease. But as you heard David say in his talk as well, there is a possibility. Hello? So, so what happens is, as you heard from David's talk as well, because the virus is there, can everybody hear me?
Yes, we can hear you.
Okay, fine. Okay, so what happens is the virus can sometimes mutate and cause differences in our bodies, but there's a lot of safety protocols in place that one should always abide by. So this is using the actual virus where we get the immune response and being produced by this by the virus as the virus replicates. So then the next slide, please, is the viral vector vaccines. This we use a virus like adenovirus. Adenovirus is a virus that does not cause disease, and what we do, we engineer a piece of the coronavirus. We introduce it into, by genetic engineering, introduce the proteins of those coronavirus into the adenovirus in our body, and these viruses then become weakened, and the adenovirus, we take it very well, is not a bad virus for us at all, and it replicates, and in that sense, the vaccines are very effective. The only sometimes drawback the adenovirus-based vaccines can have is, as we have a second dose, the vaccine can become very less effective, and also some people may have already immunity to the virus. It is this adenovirus because it's so prevalent within the human population. So, but these ones, they are very safe viruses, very safe viral vectors vaccines. In my view, are very, very safe vaccines. And those are the Oxford AstraZeneca, the Johnson & Johnson. Of course, as I'm talking about this is the basics of the whole recombinant vaccine production, but we have to also use other things like the beauty of the recombinant vaccines is that we only remove what we require from the virus. So what happens is that we don't have the redundant components that belong to the actual, let's say, the coronavirus in this case. So all these potential toxic, allergic, and autoimmune triggering components of the pathogen have been removed. However, in doing so, there's also a little bit of a drawback in the sense that we remove most of the danger signals that our body can have. So we have to use immune stimulants, which are the so-called adjuvants, and as the delivery systems. So sometimes those adjuvants can cause a problem. Sometimes the vaccines can have a problem, not because of what they have in there, but the adjuvants that they have been used to produce the vaccine. In other words, to induce a response, a stimulant of our immune system. So this is the kind of things that one has to always bear in mind. So now the next set of vaccines, please, the next slide. Those are the nucleic acid vaccines. And what they do, they use the genetic instruction, can either be the DNA or the RNA for the coronavirus protein that prompts immune system. So the whole nucleic acid is inserted into the human cells. In the case of the ones we're talking about, the Pfizer and the Moderna vaccines, in the case of the coronavirus, which is the RNA virus, so we don't have the DNA in there, the RNA virus, we use the RNA straight away. The Pfizer and Moderna don't convert into DNA, they use straight away the messenger RNA. And to prevent degradation of the messenger RNA, they just encapsulate it into a lipid kind of, is a protein, a lipid protein, in order to be able to get into the virus. Now, some of them can be nanoparticles, can be a variety of encapsulations that we can use. And there can be some controversies about it, but of course there's been some, they were approved for emergency use. So this is what it is. The whole virus is in there. And the viral protein is in there, the spike protein is in there, and we produce the virus. The body produces the virus. Then, so many, now just the next slide, please. The protein-based vaccines, they use portions, fragments of the proteins, or the protein even shells of the coronavirus outer membrane that mimic the coronavirus, and then we produce an immune response. So these are also pretty, and they inject directly. These are also pretty safe vaccines in a way, and they've got the virus-like particles too. The virus-like particles, they require a lot of adjuvants because, and those are a bit expensive to produce, but all of these require adjuvants. So in the next slide, if you can just see, the vaccines in development, as you can see, there's an array of them, and we're all producing all of these because we still need to understand them. We still need to see what the effect is going to be in the worldwide population. So basically, we have to be alert and hear what are the outcomes of these vaccines all over the world. And because actually we in a way are being the trials, the world trial is a kind of the final trial is being the world trial, where you can see which are the most effective ones amongst the different populations of the world, because we are not all the same. Our genetic makeup is not all the same, and we react differently. We are open to different environments. Our environments, we produce different, our immune system is stronger in some ways or another. So that is why we're going to be reacting differently towards the vaccines, and we need to have it evaluated all over the world. So when I hear that Europe, America, China, everybody's having the vaccine, it's actually quite okay for us to watch and see what is happening before we actually take over a lot of those other vaccines. So as you can see, this is the kind of vaccines that are being produced worldwide. So now for the next slide, please, is the vaccines for SARS-CoV-2 variants. The variants is something that we can expect. Everybody expected variants because as we try to be smart with our vaccines and try to overcome the virus is not sitting still. The virus is also smart. So the virus is also trying to overcome the problems we're giving into the vaccines. So variants appear. There's many reasons, many different ways our variants to appear. So the first wave of vaccines, we targeted the S protein. Now, the next generation of vaccines, they need to go for a broadened target of viral antigens to protect across strains. That it will enable us to be for the vaccine to be a bit more long-lived, and also to avoid different, as the previous speaker of mine said, what are we being vaccinated against? Because it's exactly that the variants are coming into the world. So the next generation of vaccines, when they look, and they're already being produced anyway, the different viral epitopes, not the S protein anymore, which are more conserved ones for the virus epitopes that elicit a broadly neutralizing antibody responses, then I think in that way we will be a little bit ahead of the virus. But as the previous speaker said as well, we don't just have to wait for the vaccines. if our immune system is strong enough, we should also be able to cope with what is coming. But this is not the point of this. What I'm trying to talk about is this is how the vaccines are being produced, and vaccines for coronavirus will have to be produced all the time because the variants are coming all the time. So we have to target the conserved epitopes of the virus. And the next slide, please. I think I'm done. Yes, and I'm done.
Please unmute, sir.
Let me unmute. I don't know my video. I can't see.
We can see you though.
Okay. Okay. Thank you so much, Dr. Eugenia Barros, for the comprehensive presentation you have made. Indeed, this is a learning day, and we will emerge out of this webinar better than before. Without further wasting more time, let me welcome our last speaker for the day. He's a PhD holder from Columbia University. Bruce Melado is a full professor at the University of the Witwatersrand, WITS, a senior researcher of Itemba Labs, and serves as the director of the Institute for Collider Particle Physics. Prof. Melado is the national contact physicist of South Africa at the Atlas Experiment and CERN, and is the co-chair of the Nuclear Particle and Radiation Division of South African Institute of Physics. He is the recipient of several awards and fellowships. Prof. Melado is an internationally acclaimed B1 rated researcher of the National Research Foundation of South Africa. Prof. Melado is an expert on the Higgs boson, a subatomic particle that is thought to give matter its mass, and was a leading participant in its discovery that was announced in 2012 and led to the Nobel Prize in Physics being awarded in 2013 to Francois Engels and Peter W. Higgs. He has attained and continued to have a number of positions of leadership internationally. Prof. Melado is a member of the Gauteng Premier's COVID-19 Advisory Committee, where he leads work on predictions. Prof. Melado is also the co-president of the Africa-Canada Artificial Intelligence Data Modeling Consortium. The project received grant awards by the IEEE and the IDRC, and also includes partnerships with IBM and Amazon. The project has been covered extensively on TV, radio, and major newspapers for a selection of interviews and articles on the press. Prof. Melado, it's now your time. Forgive me for reading like I'm a former President Jacob Zuma, but you may go ahead.
Can you hear me? Hello, can you hear me?
Yes, we can.
All right.
I can on my side.
Introduction. I will be talking about the work we've doing for the past few months in partnership with the Office of the Premier, the Department of Health of the Gauteng province and other stakeholders. And as the already chair indicated, this is a project that's funded by the Canadian IDRC, the American IEEE, and in partnership with IBM. And I'll be basically focusing on what we made public to the population on Friday through the Office of the Premier and the press conference we held on Friday. All right, so as a quick reminder, I think everybody's familiar with the setup here for the rollout of the vaccine. We're currently in phase one. Phase one is intended to preserve the integrity of the healthcare system by vaccinating healthcare workers that are most exposed to the virus. That will be followed by phase 2, which will include the rest of essential workers and populations at risk. That is phase 2. Phase 2B is where we deal with the big numbers, where we have a large number of people with comorbidities or certain risks. And that's where data science comes in, and that's specialists like us come in to help. The intent of phase 2 is there in order to suppress the probability of severe disease, number of people going to ICUs or dying. So that's a very, very important phase that once achieved will lead to phase 3, which is intended to cater to a much wider constituency in order to achieve population immunity. So each phase has a role and each phase has a prioritization. What we have done in the province through the advisory committee that I'm a member of is to devise a fine-tuned version of the prioritization policies for phase 2, for which we had to be prepared, and that's what was announced on last Friday at the press conference of the Premier. So what is the approach? What approach did we adopt in order to deal with this problem? Clearly, this is a multidimensional problem with many factors that includes the probability of a certain person to contract the virus, so the level of exposure, the probability once having contracted the virus of contracting severe illness, the existence or not of antibodies, social vulnerabilities, and so on and so forth. So we developed a project in collaboration with York University in Canada, funded by the Canadian IDRC, to basically feed all this into a machine learning algorithm, that's artificial intelligence, in order to help policymakers deal with prioritization. Now, this is all great and fantastic. This is obviously the work of scientists, and obviously the reality on the ground and the ability to connect with the policymaker obviously forces us to simplify the approach. And that's what was adopted within the context of the work stream that was put together by the Office of the Premier and the Department of Health at the level of Gauteng to define priorities and who should be able to get it first in order to have the biggest possible impact of limited vaccination. So as a result of lengthy discussions and few months of work, we basically concluded that in regard to the rollout of phase, and in particular phase 2B, we should take a two-pronged approach, a simplified version of what I just showed in the previous slide. The one prong or the one aspect that needs to be dealt with in order to incorporate the WHO value framework is in order to create a vulnerability map in terms of social vulnerabilities that can affect outcomes in this pandemic. For instance, inability to social distance because the family lives in a very small dwelling, the fact that the person has to use public transportation, and things like that. These are basically factors that enhance the probability of contracting the virus or developing severe disease. And then in parallel, the second prong would be about creating a smart algorithm that would basically tell us what is the probability of an individual, given age, given comorbidities, to develop severe illness. So this basically puts together the two types of vulnerabilities. One is the socioeconomic vulnerability, and second is the pure individual vulnerability, which basically is linked to a probability to develop a severe illness. Let me talk about the second now first, and I will end my presentation on the first issue, the social vulnerabilities. So first, we're going to introduce artificial intelligence in order to model the individual risk of a person to develop severe illness, given the data that we already have and look and study. So what we do is we take multidimensional data from the Department of Health that includes age, race, gender, comorbidities, type of hospitalization, whether the person was hospitalized in general ward and discharged from there, or the person was move to ICU, high care, and discharge alive or dead. So there's a bunch of parameters that the data provide. And what we decided to do, and I don't have time to go into the methodology and why the methodology, but what we decided to do is to create a model for severe disease that would include the characteristics of patients that made it to the ICU, high care, or died. So when we say ICU, high care, there are those that actually were discharged either dead or alive. And actually what we saw is that there is, among those that made it to the ICU and high care or died, or were discharged from ICU alive, or were moved from ICU to another ward alive, there are not very serious differences in terms of the comorbidities, age, and so on and so forth. So it made a whole lot of sense to put everything together in order to have a less noisy modeling building for the point of view of the statistics and the mathematics. So then you separate the samples into training and testing and use methodologies in semi-supervision and artificial intelligence to create a model. Then that model, and I cannot go to obviously all the technical details, is compared with the actual population, which has now a more realistic distribution and model of comorbidities in Gauteng and the age, and that comes from the population surveys that the GCRO, one of our partners, has done in recent years, basically giving us the door on the way to calculate the probability of a citizen in Gauteng to contract severe illness and or die. So this led to a smart algorithm. This is a slide that was shown by Dr. Mary Kawonga, who is our chair, the chair of our advisory committee, which has now also been adopted as a baseline for a fine-tuned approach to vaccination by the province, and this was officially presented, as I said, on Friday during the Premier's conference. And what this graph is telling you, this blue curve is telling you, is that by vaccinating about 20% of the population, you can achieve about an 80% reduction of severe disease, assuming, of course, that the vaccine that will be chosen is highly efficient against severe disease. Obviously, one has to distinguish here between mild disease and severe disease in the way we have defined it. So basically, this is my algorithms tell you that as we should be able to have a very big impact with a relatively small number of vaccines if those vaccines are distributed in a smart and efficient way. If you look at the age profile, what we decided to do is we basically created 5 different types of groups, population groups, based purely on age, gender, race, and comorbidities. So it doesn't really take into account the socioeconomics that will come later. And you can see from the most risk, that's the first graph, to the least risk. You can clearly see that the age plays a very important factor. Basically, people about 60 years of age or more certainly have a very high probability of passing on. But as we will see in a minute, it's not only age, and that's really where artificial intelligence comes in to help. It's not just, as you see here, it's not just age, but also the number of pre-existing conditions or comorbidities that are relevant to COVID-19, such as hypertension, heart disease, and diabetes. Those are the main 3 factors that lead to fatal outcomes or severe disease in COVID-19. And you can see people with highest risk are not only with age 60 and above, but have at least 2 comorbidities. That's extremely important in order to really decide which way to go in terms of how to vaccinate the population. So in terms of that, this is an official slide from the province. This is a table of risk groups in terms of age and comorbidities and the fraction of the population that entails over 18, which basically gives you a measure of how many vaccines would you need in order to cover risk group 1 and then reduce mortality through risk group 1 and so on and so forth. Because it's very important to basically, instead of just giving to policymakers the output from an artificial intelligence algorithm, you really need to have a very clearly stated, compartmentalized advisory that can be easily implemented. So in order to arrive at this classification in terms of age and comorbidities, we use artificial intelligence that underlies and underpins this table. And this was, in fact, this was shown on Friday. Now, let's talk about social vulnerabilities, because we cannot, in a society like South Africa with such stark social differences, we can't ignore the subject of social vulnerabilities in the context of the rollout of the vaccine. And there's been a lot of discussions about how to implement this in a relatively short time scale in order to be effective. Because what we don't want to do is to basically lose ourselves in endless discussions with wishful thinking and very good ideas, but they're very difficult to implement. And in the end of the day, we arrived at a baseline approach that allows us to guide policymakers in the process of administering the vaccine in order not to leave the most vulnerable behind. So we need to basically classify and establish levels of risks from the social point of view. So in collaboration with our colleagues at the GCRO, we've created this classification of risks intertwined with population density. So we have here it's a two-dimensional graph. On the x-axis, you have population density. As we know, population density plays a very strong role in the spread of the virus, but it's also strongly correlated with social vulnerabilities. On the y-axis is a combined vulnerability index that puts together a bunch of other indexes that are relevant to to COVID-19, for instance, the ability or inability to social distance because of where you live, the access to running water, and things like that. And then we've basically created a quadrant with 4 different levels of density of risk, 1, 2, 3, 4, that would correspond to each of the quadrants of this 2-dimensional graph. And then all the 529 words in Gauteng have been classified accordingly. This has been put together by our GCRO colleagues in a map. And you can see basically they're following color codes, red being the most vulnerable, then orange the next stage, and the lower vulnerabilities in blue, dark, and light blue, indicating the different levels of risk driven by social vulnerability. So it's very important, and when it comes to managing this complex problem, it's very important to visualize. And it's very important for policymakers to actually have a clear picture, or on the basis of an objective mathematical metric, to say these are the areas that are most vulnerable based on the critical factors. And then they can really visualize where those areas are and ascertain that the distribution of the vaccination is equitative from that point of view. In other words, that the vaccination process, which is a very convoluted one because it takes into account not just the availability of the vaccine, but also how to administer and where to administer, does not leave the most vulnerable communities behind. So now we have a very clear picture. In addition to that, in collaboration with our colleagues from IBM who have done this work for pro bono, they've developed an interactive map, a website, gpcoronavirus.co.za, where all these factors can be visualized through an interactive map. People can zoom in, can zoom out, can look at different parameters, the index of risks, the different kinds of risks that that compound index is composed of, the amount of population, the amount of vaccination that's required, et cetera, in order to be able to have a visualization of this complex problem that includes both the risk at the individual level and the social vulnerabilities at the same time. And that is basically the baseline that the Gauteng province has adopted in order to roll out and oversee the rollout of the vaccination in the province. Thank you very much.
Well, Prof, I believe you are done.
Yes, sir.
Okay. Now is going to be a time for the question and answer session. And in no particular order, there are no pre-submitted asked questions here. So I'm gonna ask that I work with the admin in identifying everybody who wants to ask a question. And I'm not sure on the chat if there are people already who have asked specific questions. It will help for our guests to look at the chat as well to check if there are questions that are being asked. So I will, by show of hand, may I identify the people who are going to be asking questions. I'd really love to limit the number of questions because we are chasing time, but we still have a little bit of time because this session is scheduled to conclude at 2 o'clock. So if I may be assisted, I can't see anything on my side. If I may be assisted, if there are people who have asked questions. I can also see hands.
There seems to be no hands.
Oh, there seem to be no hands.
Yeah.
Okay, so can I assume this was clear and precise? The whole country is aware that where we are going with all these issues. If there are no hands, no questions, means this was spot on and clear. So that's my assumption now.
Mr. Program Director, maybe I could just ask Bruce a couple of questions. So I was just surprised that bronchospray comes up as a high-risk area, because my understanding is that in terms of population density, it's out in, in the countryside, as it were. So it just struck me. And then the other question I have is, okay, so you're modeling the need for the vaccine, right? But have you also then modeled the workforce that is required? So in other words, what are the places that would be required? Are you going to be using churches, pharmacies, clinics? And then what workforce would you need to be able to do the vaccination as quickly as possible?
Okay, can I take the questions, Chair? Hello.
Yeah, I think you can go ahead.
Yes. Okay. All right. So in regard to your first questions, bronchospray, okay, we can look into it in detail, but essentially we're using official stats. It's a combination of the surveys plus SAS stats that give fairly precise understanding of the parameters we use. But I mean, I'm very happy. I will forward this request to the technical team to look into to ascertain that in fact no bug happened in the code. But the reality is that we use officially, official stats there. In regard to the modeling, so you have asked a very good question there, and in fact, this is now the big next step. Now that we've established a fine-tuned version, I think I would say a fairly detailed plan of vaccination, the next step now is to understand the capabilities to roll out. Now, we had a conversation, we had a group, a team meeting about that last Saturday to basically move to actually address that question. We do have information from the Department of Health in terms of how many stations. I think it's about 2,000 stations available. How long does it take to vaccinate a person? How many vaccinees can a worker deal with? And also the corresponding bookkeeping to take place and all that. So all those estimates exist. But of course, obviously, we can't relax and assume that because we sort of know the picture, roughly speaking, that everything is okay. So we are now in the next few weeks, while we're still waiting for a large number of vaccines to run simulations in collaboration with our colleagues in Canada who have done it already in the province of Ontario, to basically introduce some realism in the modeling in order to understand potential pitfalls that could lead to certain inefficiencies that could decrease the amount of vaccinations that can be provided. But you certainly ask a very good question. That's really going to be the focus of our team in the next few weeks in order to really end up having I would say, a realistic but also fairly robust, mathematically speaking, estimate of how many people we can vaccinate today in the province of Gauteng.
I guess there's a question from Paul and then from Dr. Russon. Maybe they can just ask because they've got their hands up.
All right, we'll start with Paul. Paul, fix your mic. Okay.
Yes. Good afternoon, everyone. My question is very simple. I watch a CNN program where a family created a group around the after effects of the vaccine. In their mind, they were just expecting to have few people. The group now, it's well over 70,000 people with after effects. My question then says, is the various vaccines that are about to be administered in the country, are they safe in the short term, medium term, and the long term? Thank you.
All right, maybe I'll take that question. And like I said in this talk, nothing is safe. And so, well, look, they're as safe as the evidence that is currently there. And when you talk about safety, it's when you talk about side effects. So we have what are things that are called side effects, which are things that you would kind of expect. So for instance, if you take the vaccine, you might have pain at the site of injection. You might, I've heard of, say, you know, people who feel very tired. And actually that is expected. You might even be feverish. And that is expected because your immune system is now kicking into gear, as it were. So if it's a 2-dose vaccine, your second dose is probably going to have more side effects than your first. Because remember, your first is just to prime the immune system. Then the second one is almost like you're now testing the immune system. So you are assaulting the immune system and expecting some kind of response, right? So those would be normal side effects. Then we have what would be called adverse events, or and these are might be fairly more severe, right? But you also then have to weigh the risk, what we call risk-benefit ratio. So the risk of dying from corona is real, right? But then the risk of dying from the vaccine is very small. So those are some of the considerations that you need to take when you have these vaccines. I'm sure that your children have been vaccinated and they are okay. You know, they had the odd temperature, you gave them paracetamol and they're okay. And then nothing does not have a side effect. It's the same with water. If you drink too much water, you are probably going to have, well, the side effect is that you go to the toilet a lot. But then in terms of adverse event, you could potentially have, you know, kidney problems coming in. Thank you.
Hello. Can you hear me, facilitator?
Okay, yes, I can hear you. Am I right to say Dr. Russon has a question?
That's correct, yes.
Yes, may you please go through.
Thank you so much. My name is Dr. Rasin. I'm a researcher and sociotherapist. I'm also a leader of a political party called Shosholoza Progressive Party. We're very interested in the subject of the vaccine. My first question goes to Professor Bruce Millardo. He talks about a vaccination program, but our information is that there is no vaccination program in South Africa at the moment. The current vaccination that is being undertaken is a test or a trial. Our government is using our taxes to assist Johnson & Johnson to trial its vaccine on our healthcare workers. And they've vaccinated or trialed over 100,000 healthcare workers on this untested and unapproved vaccine. We believe this is a high risk, but Professor Bruce presents it like it is a vaccination program. Parliament questioned the Ministry of Health about this because it's dishonest to tell the public that we are vaccinating when we are testing a vaccine. And this vaccine has not been approved yet. It's under trial. So that needs to be put in place. And what amazes me also is that this vaccine is moving from a vaccine against COVID-19 to a vaccine against severe disease. Things like hypertension, diabetes, cardiac disease, and all that. Isn't it supposed to be a vaccine against COVID-19? The nice thing about this corona pandemic is that it has exposed the holoprinted of our science community. Scientists have been exposed to be not just dishonest, but manipulative and liars. They've been intimidating us and telling us of figures that hundreds of thousands of our people are going to die because of the COVID-19 pandemic. And then it mutated in November last year. There's a new variant. And how then do you vaccinate us with the COVID-19? Johnson & Johnson vaccine that uses the viral vector, which, let me explain so that we can understand what the viral vector is. They take the COVID-19 virus and insert it in another virus, like an egg. You put in the COVID-19 inside the egg, it's the yolk, and the outside shell is the vector that carries the real virus. We wrote to the Minister of Health, finding out what is this a vector that is being used to carry the COVID-19. We wanted specifications, and they have not responded to us. Because our studies have showed that if the vector is a virus that the population is already immune to, like you're using measles as the vector to contain COVID-19, then it's not going to work because we are already immune to measles. That information has not been given to us. So there's a lot of manipulation, there's a lot of lies going on here. And we suspect that what the first presenter said holds a lot of water, that the return on investment on vaccine is very high. And Bill Gates has himself actually acknowledged that his best investment has been in vaccines. He gets the most money in investing in vaccines. So how true is it that indeed this J&J vaccine is going to work? They are changing the stories. It was supposed to be a vaccine against COVID-19. COVID-19 mutated to something else. And now they say it's about severe disease. Don't they think we're stupid? No. We cannot deal with this nonsense anymore. The next question I want to ask is about, it's to Eugenia Barros. She has not told us that we have selected the viral vector, which is the Johnson & Johnson, over the DNA RNA vaccine. And why have we done that? And why are we not giving the actual information about the two viruses that are being used in the viral vector used by Johnson & Johnson? We need to know that information. We are entitled to that information. And to the first presentation, I want to ask a question that might sound very stupid, but it's not, because we're looking at it. Do viruses really exist? Google that question. Do viruses really exist? There is a new thesis that says viruses don't exist. The body produces the protein, the various protein, using its instructions from its RNA to combat infections and changes in the atmospheric environment. And then we take the tissue from the human body, we analyze it, we find a protein, and we say that protein is a virus. A virus is defined as a lifeless entity that goes into the human body and it hijacks the human protein to survive or to make a life for itself. How true is it that this thing exists without a life of its own? And how does it then move into the human body if it doesn't have a life of its own? These are the questions we need to ask. Do viruses really exist? Germs do exist, yes, because they have a life of their own. But if a virus doesn't have a life of its own, does it really exist? Thank you. I'll stop there.
Thank you so much for the thought-provoking questions that are here. May I indulge our speakers to look into the chat box for additional questions that are being asked. I see the last one came from Dr. J.S. Dirani as well. Please familiarize yourself with the questions on the chat box. We've got now a few more minutes to go before we close this webinar session. May I then ask our speakers in no particular order to answer the questions, or maybe just for to do this, I'll do it backwards now. I will start with Professor Bruce Melado. If he has got questions to answer, this is now your platform. Then I will come to Dr. Eugenia Barros. Then I will end up with Professor David Katereri. Unless Mr. Maponga is back, I will ask him also to respond. But let's start with Professor Bruce Melado.
Okay, so first of all, thanks for the comments. I will address what I can address from what Dr. Rusan has indicated. So as a member of the advisory committee, I cannot and I will not make any statements or comments on political issues. What our mandate is in regard to the vaccination rollout is to establish a well-defined plan for phase 1, phase 2, and phase 3 of the vaccination rollout, as already described by National Department of Health, and how to implement that within the context of Gauteng, taking into account the specifics of Gauteng and fine-tune it for Gauteng to enhance the efficiency of the rollout. And all the phases pertain to the fight against COVID-19, whether it is phase 1, which is basically protect the integrity of the healthcare system by first inoculating the healthcare workers. Phase 2 is more about targeting the rest of the of people that can easily be exposed to the virus, and sectors of the population with age and or comorbidities, in order to reduce severe illness with a relatively small fraction of the vaccines, and then move to phase 3.
It's a trial, Bruce. It's a trial. It's not a vaccine. You are trialing it. Be honest. It's a test.
This
you want me to quote the...
Sorry, can you let me finish, please? This is in regard to the entire phase 1, phase 2, and phase 3, how to prioritize on that basis, regardless of the trial aspect of it. This is basically a plan to ensure that when the vaccines arrive, we know what to do, how to do it, and who to inoculate in order to be most effective. And that's really as far as I can go in terms of answering your questions. Thank you.
Can I just take my questions now?
Yes, yes, please.
Okay.
You may go ahead. You may go ahead, please.
Yes. Okay. So regarding the use, the Johnson & Johnson vaccine and the use of adenovirus, we don't take the coronavirus and push it into the adenovirus. We take a piece of the genetic material of the coronavirus and we introduce it into adenovirus because as a vector, and the adenovirus will not replicate in our body. So it's not one virus into another virus, a piece of the virus protein that will now be expressed in adenovirus. The issue about adenovirus being a very common virus amongst the population, it's reality. But I guess because it's a very safe vaccine, I guess the point is to elicit some immunity. So even if it's not 100%, it's to elicit some immunity towards people that may be getting the infection of the coronavirus and become very ill. Now, regarding the RNA vaccine, the Pfizer ones and the Moderna, they just take the full RNA. And in some cases, just take a portion of the spike protein and they engulf it into this lipid liposome section and sometimes put the nanoparticles and they insert it into the body. And then our body is supposed to be producing the viruses and in that sense, produce immunity. So it's completely different technology. And the Johnson & Johnson is a well-known technology. The Moderna and the Pfizer is a brand new technology. So that is why we have to monitor worldwide what kind of reactions we can have to this.
Right. Yeah. What I think would like to say is that so pathogens have pathogenesis, and they would show up with certain symptoms. Right. So this actually reminds me of the old debate around HIV and AIDS. Right. So the thing is, we know that there's a pathogen, however you might want to call it, and that this pathogen has typical symptoms. So when you get it, you have loss of taste, loss of smell. Then you might, if it complicates, have breathing difficulties and leading to death. So there is something out there. Right, and I think the scientific world is agreed that it is a virus, because it can't be a fungus or a bacterium. And then maybe on the trialing of vaccines, let me maybe just say to you that there actually isn't a single country in the world which has fully approved these vaccines. All these vaccines are being brought into countries using what's called emergency use approvals, right? So we are in unprecedented times where we have a global pandemic. So the issue is that, as far as we know, the vaccines have been tested for safety and for quality, to a point where we are confident enough that they can be introduced into human populations. And this is being done through what's called emergency use approval. And then what you do have, like with everything else, with paracetamol, with aspirin, you then have what's called post-marketing surveillance, which is why I was talking a lot about users need to be reporting side effects and adverse events, because otherwise we could wait for a long time while people are dying. So again, it goes back to risk versus benefit. At individual level, risk versus benefit at population level. Thank you.
Well, it looks like we have come to the end of our program, and thank you very much, ladies and gentlemen, for participating in such a very important Zoom webinar program where we discuss the historical challenges and opportunities with vaccine development. One of my former professors who was the council chair of a historic university in South Africa that used to be called Vista, Professor Ngabi, once said, people who do not know where they are coming from end up arriving where they are coming from and think that is their destination. With these words, I would like to thank everybody who participated, and we are looking forward to more lively sessions such as this one. An engaging society is always a better one. Thank you to the Vice Chancellor, the Deputy Vice Chancellors, the university management, the student leadership, and the participants at large, including all the international guests that made this program a success today. Thank you so much, and until we meet again.
Thank you very much.
Thank you. Bye-bye.
Thank you. Bye. This your boy Drake. Station. 93.6. Hot new music. Just like that. What up? This is Pink. 20.